Okay. The title of my talk is Small Molecules Inhibitors of Breast Cancer. I'm coming from University of Cincinnati in Ohio. So first of all, some information on incidence and mortality of breast cancer worldwide. It's quite surprising. You can see here the cases per 100,000 people. That's the incidence. And in red, you see the mortality. You can see the US, Netherlands, Denmark, France, Australia, and all these very high incidence of breast cancer. As we go down more towards Asia and all this, you can see a drop in the incidence of breast cancer and also in the mortality. And as we go to India, Zimbabwe, and China, it's so much smaller relative to what happened in the Western world. So why am I talking about prolactin? A lot of studies, epidemiological, pathological, and experimental data have shown multiple oncogenic actions of prolactin, PRL, in breast cancer. Prolactin is considered a pituitary hormone, a 23 kilodalton protein. But unique to humans, prolactin is produced not only by the pituitary gland, but also by breast cancer cells, by adipocytes, and also by immune cells, where it acts as an autocrine paracrine factor. The hypothesis we had, and I've been working on prolactin for many, many years, is that effective blockade of prolactin action should result in the suppression of breast cancer growth, inhibition of metastasis, and increased chemosensitivity. So here were the objectives of our study that lasted about 10 years. We use a high throughput screening, HTS strategy in combination with cell-based assays to identify small molecule inhibitors, we call them SMI, of the prolactin receptor, PRLR. We then determine the binding affinity of the SMI, the small molecule inhibitors, to the prolactin receptor and examine their mechanism of action. Then we establish the bioactivity, lack of toxicity, and pharmacokinetics of the SMIs. Then we had to demonstrate the ability to suppress tumor growth in immune-deficient mice. In other words, in mice that we can implant human breast cancer cells to see how it affects human breast cancer cells. So what do we know about prolactin? There are in humans multiple production sites. It's regulated by local factors, and it has also constitutive non-vesicular secretion. That's not like many endocrine compounds. The structure of the receptor is like a cytokine. The receptor is distributed almost all over the body, and the main signaling is through the JAK-STAT and MAP kinase. So what do we know here about where prolactin is coming? The main prolactin is coming through the endocrine, through the blood, coming from the pituitary gland, and go to all over the body. However, immune cells, there is a paracrine productions, and next door cells, like adipocytes or immune cells, can affect the breast also. And then there is an autocrine prolactin production, but the breast cells themselves can produce their own prolactin. So there are multiple sources that the breast can be exposed to prolactin. What are the signaling pathways that are activated at prolactin in breast cancer? Okay. In females, you see this. These are the ones that are autocrine prolactin increases the tumor growth, and this is the vector control. So you can see it here. In females, you see the same story, and this is now the tumor growth per week. cubic millimeter per week, and you can see here the 3 different clones that overexpress prolactin grow much faster than the normal cells. And now another interesting thing, here you have one of the clone, 23K2 and 23K3, and the 2 vector controls. You can see they are overexpressing, this is Western blot, overexpressing prolactin, but they also stimulate excessive production of the prolactin receptor. So if the ligand is high, also the receptor also is activated. And we don't see that here in the vector controls. This is now the actin, BCL2, and BAX here. So what do we know about breast cancer and chemotherapy? Here you can see here how many patients are alive after diagnosis of breast cancer, and what is the treatment cost. The treatment cost goes up depending upon the type of stage of the breast cancer. This is stage 0, stage 1, stage 2, and etc. The cost increases and the survival decreases. So worldwide, we have over 1 million women that are diagnosed annually with breast cancer, and more than 400,000 will die from the disease. Chemotherapy remains the mainstay, the main treatment for patients with triple-negative tumors that don't have the ER, the estrogen receptor, progesterone receptor, and HER2, and those with metastatic disease. While the choice and success of therapeutic drugs have increased, the choice and success of therapeutic drugs have increased, the tumor resistance to chemotherapy remain a major obstacle that results in treatment failure. So we need other treatments. Because of prolactin acts as a survival factor, we question if it antagonizes the cytotoxicity of chemotherapeutic drugs. So we did a lot of studies with breast cancer cells. You see an example here. Here is the cell viability, a % of control. If you treat them with a drug alone, Taxol, vinblastine, doxorubicin, or cisplatin, you can see here the cell die as you increase the drug concentration. The same here, the same here, and the same here. However, if you give the drug together with prolactin, there's a shift to the right, and you need much more drug in order to cause cell death. You see in all cases here. So prolactin acts as increases resistance to chemotherapeutic drugs in breast cancer. Now, most treatment with the antibodies are large proteins, but we want to see what is the advantage of small molecules compared to protein-based therapeutics in breast cancer. First of all, small compounds can be delivered by just swallowing it. So the oral deliverability. There is a potential for the small molecules to penetrate into the brain if there are metastases, while large molecules are prevented from accessing the brain. They don't cause immunorejection. Because the small molecules, you can optimize their structure by medicinal chemistry. Relative to large protein like antibodies and so on, the low production cost and reduced patient expenses. So what are the facilities at the Our University for Drug Discovery Center? This was a gift from Procter & Gamble, which is located in Cincinnati. They have a small molecule library that includes 350,000 molecules. These molecules were selected because they act as drug-like compounds. They're smaller than 500 Dalton, so they're really small molecules. A lot of structural diversity, so you have a chance to discover molecules that are really going to act. They remove reactives, remove toxic compounds, the solubility. This is what the machine looks like. It's quite an expensive machine. And basically what you see that you use a 384 well plates, you put the cells that you're going to check. This way increase the drugs and you can quantitate as the cell are either alive or dying. And in the drug discovery center, you have a lot of experts in drug discovery. You have people who can do medicinal chemistry, people who can do molecular imaging, and also use bioinformatics. So what did we do to discover the small molecules? This is the screening strategy. First of all, we did a model of trying to dock the ligands to the prolactin receptor by computer modeling. And virtually we screened the library, established 3 small assays to discover I'll show you in a minute, optimize the assays, adapt to high HTS, and start screening 350,000 compounds. From the 350 compounds, we screened 51,000 compounds. And then we retested the hits and examining the inhibitory constant. We use, in a minute I explained it, the microscale thermophoresis. We use the prolactin extracellular domain. We determined the binding affinity. We did molecular binding, determined the ADME, which are the properties of the molecules, and determined the signaling. But then we use medicinal chemistry to produce the compounds and proceed. So these are the assays, and we really did this sequentially because the different sensitivity to prolactin. These are red lymphocytes that depend on prolactin for growth. And you can see here the prolactin in nanograms compounds can increase relative fluorescence unit, which is a marker of cell growth. Then we use another assay, which is BAF-F3 assay, where you need more prolactin and you get this increased cell viability. And finally, because these are murine, we needed T47D cells that are human breast cancer cells. And you can see here, they're not as sensitive to prolactin, but we need to verify when we screen with murine cells that the same thing works on human cells. And so what we see here, we screen, we use the assay, this assay from the 50,000, we reduce it to about 5,000. We use the other assay, we reduce it to about 100,000. And then we use the T47D, and we ended up with these 7 potent inhibitors. We call them SMI, small molecule inhibitors. Here is the structure, 7 molecules. And when we did the molecular imaging or the modeling, we found that even though in some of the assays they have a high I60, I50, they didn't bind to the extracellular domain of those prolactin receptors, so we did not continue. We wanted only molecules that act on the prolactin receptor. And you can see here the KD that we determined. And out of this, we selected 2 of the final 7. We call them SMI1 and SMI6. So after about 2 or 3 years of work, we came up with 2 molecules that are potentially good compounds. Now, we needed them a large amount to try them both on cell in culture and also in vivo in immune deficient mice. So our chemists synthesized one, you can see here, they started from 2 compounds, combined them, did a lot of chemical evaluation, and ended up with the structure of the SMI1. SMI6 was produced differently, the 2 different compounds, completely different, and you did, the chemists help us here, they did that, and ended up with this particular compound. So we needed to verify that prolactin and the SMI do bind to the extracellular domain of the prolactin receptor. So we did the extracellular domain of the prolactin receptor. And you can see here, this is the fracture bound from which we can calculate the KD. This is how the prolactin bound, and you can calculate at 29.9 nanomolar. And the SMI, which is SMI1 and SMI6 are very similar. The KD was 1.26 micromole. So it's very similar to many other compounds when you compare small molecules, the binding of the large proteins, about 1:40 ratio. We did in silico binding simulations. This is the receptor. This is the prolactin. And it binds to the extracellular domain here. And this is we found where the SMI1, which is this compound, where it binds about here. And SMI6 bound about here. So this was the model that we used. So now we wanted to test the ability of prolactin to induce cell invasion, lymphocyte proliferation, and JAK2 phosphorylation if they are indeed blocked by the small molecules. So you can see here cell invasion, which you use the Boyden chamber, if you're familiar with that. You put cells on a chamber and they migrate downstairs, and now you count how many cells migrated through the membrane. You can see here prolactin at 1 nanomolar increased cell migration from here to here. FBS, fetal bovine serum, which contain a lot of stimulating factor, increased similarly. If you do SMI1 alone, it doesn't do any. But if you use prolactin together with SMI1, you can see that it blocked the invasion. And similarly, if you use SMI6 together with prolactin, you do the same thing. We did a Jurkat, which is a lymphocyte, and we wanted to see how much the SMIs prevent cell proliferation. Prolactin didn't add anything to the normal cells, but when we use SMI1 and SMI6, you can see that they block it. That's suggesting to us that these SMIs work not only via the prolactin receptor, but also independent of it, which was very surprising to us. And we look at activation of the cellular activation by JAK2, and you can see here prolactin activate phosphorylation of JAK2, which is one of the downstream of the prolactin activation. And you can see also inhibition here by SMI with prolactin. Compare this to this or this to this. Didn't touch the total JAK2, but the phosphorylation, which is the activation of JAK2. And we did on densitometry with MDA MB468 cells, and you can see here that we can look at the percent inhibition. SMI1 with prolactin, 67% inhibition, SMI6 with prolactin, about 58. So we were pretty pleased with that. Now we wanted to see how it affects in vivo. So there are other considerations before we start doing it, and this is another breast cancer cell line. You can see that STAT phosphorylation increases with prolactin, SMI6 alone didn't do much of anything, but if you put the SMI6 together with prolactin, it blocks the STAT phosphorylation. Because prolactin is very similar to another hormone, which is the growth hormone, was very important. The growth hormone is so important to biology. We wanted to see whether it's affecting growth hormone. This is a T47D, which will remove the prolactin receptor. It expresses both growth hormone and growth hormone, but no prolactin. And you can see here, this is the control. Growth hormone increased STAT phosphorylation. SMI6 didn't do much of anything and did not block the growth hormone. So it attacks the prolactin receptor, but doesn't touch a very similar hormone, very similar hormone receptor, which is a growth hormone. Another thing was important is whether there is some cytotoxicity that can really be very bad if we use it for people. So we use the LDH cytotoxicity assay, and you can see here the SMI1 and SMI6 didn't increase the cytotoxicity. We look at the mice that were we injected them with SMI6. We look at the mean body weight, didn't change. At 2 different doses of the SMI6, 3 different doses SMI6. And we use a commercial screening assay of different receptor to see if it does anything with a receptor which should not prolactin. We found a serotonin receptor with some % inhibition, another serotonin receptor, and hypocretin receptor. So we have to really worry about this, but not too much. We also tested the multiple doses commercial assays against 468 kinases, and none was affected. So we felt fairly comfortable about the SMIs. So we picked up a very common breast cancer cells, MDA MB468, and we look at the growth in vitro. We wanted to see, because we had some indication that it affects through the prolactin receptor, but also prolactin independent. You can see here, this is now by overexpressing prolactin, and you can see increase in cell number. SMI6 alone, this is an SMI6 with prolactin converted. So in other words, that's suggesting to us that this has both prolactin and prolactin-independent action. Then we tested many other cells. So we noted this is MDA231, ZR751, MCF7. In all cases, we look at the increased cell number, and you can see that the SMI1 and did the same thing with SMI6 and the same thing with SMI1. It suppressed cell proliferation. And here is the plates where we grew them, and we see there is suppression of cell number. This is the control, this is cell number, and this is increased from 1 micromolar to 10 micromolar. And we did that against many other cell lines. We found the binding affinity. There's a bit problem here with the mammary epithelial cells, but still it's within about 40 or 100 times larger dose will do the effect. So if we stay with small dose, we are fairly secure. So now we transfected MDH cells with conditional autocrine prolactin, which is activated by doxycycline. And we wanted to see the effect of doxycycline on Taxol and tumor growth. You can see here when we have a doxycycline, there is increased prolactin with 1 clone, 2nd clone, and 3rd clone, D1, D2, D3. The controls don't do anything. And we can see here a resistance to Taxol. This is the control, so the increased Taxol that kills the cells. But if you use a doxycycline, which stimulates prolactin, it antagonizes the cytotoxicity of Taxol. And you can see here now when we transplanted these cells in immune-deficient mice, this is a normal. Tumor volume increases. And you can see here, I'm sorry, this is the normal, a control. And when you overexpress prolactin because of doxycycline, the tumor grows much more. So here we did an interesting study here. So this is, we injected doxycyclines to simulate prolactin production. This is the mice were transplanted with the cells that are under control of doxycycline to increase prolactin. And you can see when you give them doxycycline, the tumor volume increases very much. In other words, increased prolactin production increases the tumor. When we put azet osmotic mini pump in the neck, it produces that release SMI6, you see immediately the tumor is suppressed here. But if you do tumor growth in cells that are not transfected, you can see it here, no effect. So what do we say here? First of all, the conclusion, SMI6 bind to the prolactin receptors and interfere with receptor activation by the natural ligand. It does not prevent activation of growth hormone receptor, which is very important because growth hormone is very important to biology. SMI6 at 1 micromolar antagonizes prolactin-induced JAK2 STAT activation and suppresses the ability of prolactin to increase cell proliferation and invasion. It has high selectivity, no cytotoxicity, and it also suppresses cell growth independent of prolactin, suggests that SMI6 has both prolactin-dependent and prolactin-independent anti-tumorigenic activity. What do we know? What do we want to do in the future? First of all, to see if SMI6 inhibit other cancer cells like prostate, ovarian, liver that has prolactin receptor, determine if it acts by suppressing cell proliferation, by causing apoptosis or cell death, or by binding to similar cytokine receptors. There's a lot of work still to do. Establish a structure-function relationship, metabolic half-life, and pharmacodynamics of SMI6. Develop SMI6 as an alternative or as an adjuvant to current treatment of breast cancer. So this is, to summarize, a pessimist sees that's from Winston Churchill. A pessimist sees difficulty in every opportunity, and optimist sees the opportunity in every difficulty. This is the building where our operator was built by a crazy architect. My laboratory is here. This is the group that the people I worked with, postdoctoral fellows, students, technicians, and these are the people who are chemists that help us to do that. And the grant support was from National Institute of Health, Department of Defense, DOD, Right Cincinnati, and so on. Thank you.